There are still a few places here in front. Okay, let's start. Welcome everybody to our translational medicine cozy. It's already the fifth one, and the idea of setting it up was really to give a forum to connect people who are interested in translational and personalized medicine. And this year we were quite successful in getting actually responses. So we had 82 proceeding submissions, 6 submissions were selected for proceeding talks, with 75 abstracts submitted for the oral presentation, track and posters, and 9 were selected for short talks. And I think we were also lucky to get 3 really good keynote speakers. So it's Issacq Kohane from Harvard University, Burkhard Rost from the Technical University in Munich, and Bizan from the Institute of Cancer Research in the UK. Issacq will start, and Burkhard will be at noontime, and then Bizan will finish the cozy. As every year, we also have awards. So we have a first and second best oral presentation, and we also have a first and second best poster presentations. And therefore, you got these vouchers to select the best ones. You should go there in the poster area, which is just one floor above, between 12:45 and 2 o'clock. And the posters are the ones from G01 to G66. They're in the middle of these lanes. And then select your 3 best posters you have in your mind, and then give us back the vouchers. We also have sponsors. It's a little bit Luxembourgish concentrated. So the Luxembourg Center for Systems Biomedicine, where we are placed, the University of Luxembourg, our spin-out company ITTM for information technology for translational medicine, and also the Luxembourg Note in Luxembourg is a sponsor. And with that, I'm already finished and would like to give it to Venkata, who will introduce our first keynote speaker. Thank you.
Good morning. My name is Venkata Sadagopam. I'm from University of Luxembourg. So it's my great pleasure to introduce Professor Kohane, who is the inaugural chair of the Department of Biomedical Informatics, and Marion Nelson Professor of the Harvard Medical School. He develops and applies computational techniques to address diseases at multiple scales, from whole healthcare system as living laboratories to the functional genomics of neurodevelopment with a focus on autism. And also, from his lab, the very famous project I2B2 integrated informatics from bench to bed, and with internationally it's used by many academic health centers, and also like recently the TransMed is also combined with the I2B2, and he published several hundred papers in the medical literature, authored widely used book on microarrays for the integrative genomics. He's a member of the Institute of Medicine and American Society for Clinical Investigation. With this one, I would like to welcome further
first
ado.
of all, thank you very much, everybody, for having me. So while he's getting me set up, so by training, I'm both a computer scientist. I got my PhD in computer science in the 1980s, working in artificial intelligence and medicine. So let me tell you, as someone whose PhD in computer science was in artificial intelligence, I am fully inoculated against the hype around medicine and about AI in medicine. And I think it's important to understand it because you're going to be hearing a lot of it in the next few days. And it's a very exciting moment to be living in data-driven artificial intelligence, as opposed to the expert-driven artificial intelligence that we had in the 1980s. But what I'm hoping to see through these days is some skepticism that there's going to be some magic arising from the applications of these machine learning techniques to these large data sets without understanding a lot of the challenges that we're going to have therein. My other training was in medicine, and my PhD thesis advisor, Pete Solovich at MIT, made sure that I completed my training so I'd have credibility with the medical audiences that I'd be working with. And so although now I chair a department of biomedical informatics at Harvard Medical School, in fact, it's a very young department, only 4 years old, we do have this challenge of these two cultures, well, actually 3 cultures. One is doctors who are trying to take care of patients, who think they know what they're doing. Computational biologists who are wrestling with hard methodological problems, but often don't see the translational aspect. And basic biologists who are not that interested in the medicine and not interested in the computational techniques. And yet we're going to have to make it all work together. I mean, this is the reality when you have my gray hairs and you sort of see the big picture beyond the matrix. And so my goal in the next 40 or so minutes is the following. I want to get through my slides and make my case for you, but I want to leave time for some questions and for you to actually challenge me in some of my assertions so that this actually is a conversation. If you have no questions and you don't want to challenge me, I'll just accept that as assent, and then I'll give you back all your time so you can have more coffee before the next meeting. Agreed? All right, let's go for it. So, I'm going to make 5 points about how computational biologists can accelerate medicine, and I mean this in a very deep way. Unfortunately, every one of you will be in a situation, if you're thoughtful, you'll say, why is medicine not doing better for me, for my loved one? Things are going bad. Medicine, as it currently exists, doesn't do what we think it can do, and does not do what it could do if our ambitions together were more real. And so I'm going to give you 5, I think, rules of thumb, or heuristics, that I believe will be helpful to you. The first is, even at a late stage, even if you're my age, intensive and short apprenticeships in the medical domain are incredibly valuable. And I'm going to, and my model myself, like after my thesis advisor, Pete Solovich, spent many years after he was at Caltech and at MIT talking to doctors, really understanding what their problems were, and really engaging them in their problems. And before I go into how you do that, I want to illustrate the problem if you don't do that. And I'm going to give you this example, and I want you to think of this example as emblematic of all the problems that computational biologists that we will have if we don't understand medicine. So I was visited by a very good-looking young man. He was CEO. He is the CEO of a very successful company out of Silicon Valley. He was an MIT grad, and he had worked with Google on their knowledge map and had already some contracts with several health insurers. And he was coming to see old Zach at, by the way, for those of you who don't know me, my name is Issacq Kohane, and people call me Zach. So if you call me Zach, I know we've met at least once. And so he wanted to see what old Zach was doing. And he was talking about all the great things I did. And my competitive grad school self came to the fore. Whenever someone would come too self-confident, I'd always try to flip it over. And I said, explain this to me. This is data that we've developed across millions of patients, across dozens of hospitals. How is it that, is my cursor working? Yep. How is it if you are unfortunate to be my age and white and male, and you have a white blood count, measure of white blood cells, count of white blood cells per millimeter cubed, is low at 3 o'clock in the morning. If it's low at 3 o'clock in the morning, my chance of death in the next 3 years is 56%. And I said, Mr. Silicon Valley, good-looking CEO, how is it that if it's between 8 a.m. or 4 p.m., my chance of death with a low white blood count is only 25%. What's the difference? And I can tell you that every medical student who have asked this question knows immediately the answer to it, and 95% of computational biologists do not know the answer to this. And I just want you to think about it. It's too early in the morning. You don't have enough coffee for me to torture you Socratically. And so I won't do it, although it's one of my preferred modes of teaching. And the answer is, if it's 3 o'clock in the morning, as we used to say when I was in medical training, you're trying to die. If someone's drawing blood for you at 3 o'clock in the morning, you are dying, because there's no other reason when someone sticks a needle into you at 3 o'clock in the morning. If it's 4 o'clock in the afternoon, you're fine. You're in an outpatient clinic. You have a low white blood count, big deal. It's 3 o'clock in the morning and someone's drawing a blood for anything, you're going to die very soon. And of course, the reverse causality says, please don't draw my blood, you know. Doesn't work quite that way. But it's a good example of reverse causality. But here's the point. Mr. Silicon Valley, well-funded startup, working with Google, working with insurance companies on predictive modeling. His parents grew up in India, and they were both doctors, and so he desperately tried to find an answer. For him, the best he could come up with was circadian rhythm, which is not what it is. And the problem is, to him, what we just described is noise, unexplained variation. But of course, it's not noise. It's actually bias. But it's actually not bias. It's actually signal. Because if you actually know what's going on in the healthcare system, you can use that to actually change your prediction. And the point we make, and by the way, we published this in the British Medical Journal, I think last year. Last year, yeah. But the point is, there's really two systems. There's patient pathophysiology, and everybody, when they look at healthcare data, they think they're modeling patient pathophysiology. But they're not. They're actually modeling patient pathophysiology and what I call healthcare system dynamics, which is the number of observations, the time of day, the time between observations. If someone's measuring something every 3 minutes, you're about to die. If everything is being measured once every year, you're fine. So these are dynamics that are not being captured by the machine learning techniques we're using because they're not aware of this health system dynamics. And that's stupid because you could actually you'd be crowdsourcing the millions of doctors taking care of patients by using health system dynamics. You following me so far? Great. And obviously, those of us who are computer scientists can say, well, why don't we use both? Why do we have to only use one or the other? And that answer is absolutely right. But just to give you a sense, as we published in the British Medical Journal, if you look on the y-axis, it's the contribution, the informativeness of the test, and the y-axis is the informativeness of the test, and the x-axis is higher mortality versus higher survival. And what we saw was that for, I think it was 63% of the test, just knowing that the test was ordered and when was as informative as knowing the actual result of the test. And by the way, all this analysis was done by Dennis Aniel, who's now at the Rand Institute, PhD in biostatistics, not a doctor, but he bothered actually looking into the data and had this intensive apprenticeship where he actually learned the medical problems. That's the meta message I'm getting to you here. And of course, if you use both the lab value, the actual value of the lab, and the health system dynamics, you know, if you take it turns out when the Red Sox in the United States were winning the World Series, we had cured heart attacks because no one showed up in the emergency room. And so if you take these into account, these are real signals or ways to debias the signals. But if you ignore them, if you ignore the healthcare system, you are going to not have big impact because you're not looking realistically at the message. And the best PhD computer scientist, computational biologist I know, like Pete Solvich, have actually bothered understanding the domain. So, I'm going to give you some case histories. So, I can barely read this, so you probably barely. So, this is an individual who had a Bachelor of Science in Computer Science, Computer Engineering, Electrical Engineering, Stats, and Bioinformatics. And I got a note when he came for his postdoc from John Doyle, who used to be at MIT, saying this was the best grad student I'd had in the last 20 years, which was great, but the guy had zero experience in medicine. And the reason he came to work with my group is because he wanted to get immersed in medicine. And so what happened is two things happened. He came because he wanted to revisit the old goal of AI, which is automated diagnosis from the 1980s, but revisit it using embeddings, taking embeddings of healthcare, electronic health records, billing data, and all the literature of the world, using those embeddings to then deep learn the relationships of those embeddings to labels such as the ultimate diagnosis of patient in the EHR, labels such as the chapter heading in a Wikipedia document, or the discharge diagnosis in an EHR. And he learned very deeply in medicine what was going on. He did another thing, which is a useful hack, which I did not include in my 5 recommendations for computational biologists, but he married a doctor, which was very helpful to him, because his doctor wife is a pediatrician, and so he's now taking over all of deep learning on these, both the images and the content of all this neonatal data that is so underexploited in medicine. And he has published lots of papers. These are all the papers after his PhD, that when he worked with us. And as a result, he has a deep understanding of what the gritty underpinnings of medicine are, what are the right questions, what are the nature of the data. And so his name is Andrew Beam. And for those of you who've been at Neuro IPS, you know who this guy is. And he's now an assistant professor of epidemiology at the Harvard Chan School of Public Health. So that's what I call a deep apprenticeship. I'll give you another example. This individual, he has a Bachelor of Science in Physics. His PhD is in Bioinformatics and Integrative Genomics. But he became very interested in disparities across populations. And as you'll see in a later example, he did something that was not at all, that was so obvious to statistical geneticists that they would never look at it, but so unobvious to doctors that they totally missed it, with huge implications for medicine. And so this individual, Harvard MIT trained, basically a quant, also published a lot of papers after his PhD in this area. His name is Arjun Man Rai, and Raj is now a professor of, assistant professor of pediatrics at Children's Hospital and Harvard Medical School. So these are individuals, both very computational, who have actually said, I need, as part of my training, deep immersion. And it was basically a 2 to 3 year immersion, and they are now truly independent researchers with their own grants, and they're able to ask questions as well, often better, as anybody in medicine. But they had to do this training. The next, so number one rule is get immersed in the domain, so you understand what the real questions and problems are. Second is crossing the chasm between the clinical versus the computational culturally, and that means collaboration. Let me give you an example. This child showed up and had been fine. And he stopped walking, and he had been speaking, and he stopped speaking. No one understood what was going on. He was then referred to a network, which I can talk to you about in the questions section of my talk, which I hope we'll still have. Is our clock here? No. Is it really 10:40? And I have till 11:10? All right, good. So the Undiagnosed Disease Network is a network that I have the privilege of running the coordinating center of. And it's been so successful that it's gone from 7 academic health centers to 12 academic health centers. Any patient can be referred to this through our Harvard Center. All the data gets pushed into the Amazon cloud, fully identified with genomic data. So when the patient shows up, all the genomic data is there, all the clinical data is there. Never happens in medicine, but it happens in this network. And we refer them to the right doctor. And all this network, all this data is in our cloud, and we now have these 12 academic health centers. And we've seen thousands of patients. He's just one of them that was referred. So he was referred, and he was diagnosed with a deficiency of a gene I had never heard of, GTP cyclohydrolase. Now, it turns out this is very extensively expressed in the brain. And even my medical education did not know about it, but it's very important in converting the substrates that become the neurotransmitters into those neurotransmitters. So if you have a deficiency of this gene, you'll be making those neurotransmitters ineffectively. So what's happening, he was able to keep up with his brain's demands for the first few years and then no longer. So what happens, we made that diagnosis, which, remember, is a big deal because each one of you, if I look at your genome, unfortunately, every one of you is going to have about 40 to 50 mutations that, if I looked in a textbook, are actually causative of Mendelian disease. And so it's pretty tricky to figure out which one is actually causal. And it's easy to pick the wrong one and then have no effective treatment. But because we picked the right one, we treated it with actual substrates past the blockage, past the deficiency. So we gave L-dopa, folinic acid, and 5-hydroxytryptophan to this patient. And guess what? Within weeks, this kid started walking and talking again. And in fact, so well that parents were no longer used to having a young kid. They'd forgotten what it was like to have a young kid walking and talking, especially the walking part. So they just let him walk around and hadn't child-proofed the house. This guy, he fell down the stairs and broke his legs. But that was actually a good thing. It was an example of our success. But here's the point. As we've seen thousands of patients, but early results were published in the New England Journal paper, where we made the following very, very important points. That 30% of the patients that we had already referred to our network, 30% of them already had genomes or exomes sequenced. So the magic here is not in the genomic or the exomic sequencing. It's part of it. But I'm telling you that 30% of these kids, these are individuals who have been looked at. Think about you as we also see adults, but think of it for the kids. Half of these patients are kids. The parents are doing anything to solve this case. They send them throughout the world. And 30% of them had genomes and exomes and were not diagnosed. And why is that? Because either it was just a purely genomics computational team that didn't know how to sort through the mutations and figure out which mutations made sense for the clinical presentations, or they were purely clinical teams who could not actually do the appropriate analyses and actually figure out which were likely to be called out because of coverage issues or were unlikely to actually cause a coding change, for example. By the way, ironically, even at this early stage, 11% of the cases were made by clinical diagnosis alone. So that's outside the message I want to give you, but what it tells us that these patients, after seeing many doctors, just having the right doctor see them made a difference, which is really sad, because it means that if you see the wrong doctor, and these are and these are not one doctor, seeing multiple doctors, and you don't see the one who actually understands the problem, you're not going to get the right diagnosis. And we continue to see this again, and we have now achieved for these patients who've been resistant, refractory to diagnosis, we've achieved meaningful diagnosis rates of approximately 30%. So if you're a cup half empty guy, that sounds terrible, but if, like me, you're a cup half full guy, 30% is great, because these 30% of kids and adults who had never had a diagnosis despite multiple efforts. These are meaningful diagnoses. And we, by the way, we have a zebrafish core and a fly core so that we can actually CRISPR knock in the mutation to see, in fact, if they cause the phenotypes that we expect based on the clinical findings. Okay, so that's one part of my cross the chasm. Here's another part of my crossing the chasm. So hypertrophic cardiomyopathy is a disease that you actually all know about, because if you follow basketball, you've seen a few basketball players drop dead on the court because of a heart disease, and they're young men. And this heart disease is hypertrophic cardiomyopathy, where you have this abnormal growth, you get obstructed flow, and these individuals drop dead. Now, if you read a textbook when I was a medical student, and if you read the textbook today, it says prevalence is 1 in 500. Keep that in mind. And autosomal dominant. That means if you have one copy of the mutation, you have the disease, which means if you're diagnosed with it, not only are you diagnosed, but then we look at your entire family, and if you have if any family member has one copy, it's autosomal dominant, we're going to give you a workup that includes an echo, and it includes sometimes putting a defibrillator in to prevent an abnormal arrhythmia. So Arjun Manrai, the second immersed postdoc I told you about, looked at a big exome data set called the ESP data set from the National Heart Blood Institute with 6,000, it's a small data set, 6,503 individuals. And he comes back to me and he says, Zach, this is a 1 in 500 disease. Why am I finding the mutations that in the textbooks are causing this disease, autosomal dominant, in this huge proportion of these patients? Not 1 in 500, much, much higher. So I had only one useful thought to share with him. I said, go look at the race of these patients. And first of all, so these are the frequencies of these different mutations. Each of the mutations in the textbook are, quote, pathogenic, highly penetrant. And even 0.12 is much, much higher than 1 in 500. But then if you look at the race, all the signal is coming from most of the signal is coming from the African Americans. So I said, Raj, what the hell's going on here? Why are these African Americans having greater than 25% penetrance? And I don't have time to explain to you exactly how it happened, but in short, although a statistical geneticist would say, of course, there are variants in a African population, because since we all came out of Africa either as slaves 300 years ago or the rest of us 60,000 years ago, but that's the big mother pool of gene variation. And the rest of us in Asia and Europe are a tiny sample of that variation. So that if you have so what happened, if you were in Paris, as the studies were done, or in Boston, you followed the guidelines exactly, and you said, we're going to get race-matched cases and controls. So you made sure that the controls were white as can be. But you weren't quite as careful with the cases, because the cases were hard to find. So in Paris, if your grandfather or father was Algerian, guess what? Algeria is part of Africa. And so if you just fainted on the basketball court for whatever reason, and you come by and go to the doctor and you have a thick heart, which, by the way, just being an athlete will give you a thick heart, and you find a variant, that African origin variant, is unknown in white people, so it'll come out as very significantly associated with the disease. To make a long story short, because it's actually a really interesting story, but I can't devote enough time to it because I'm giving you the big picture. The statistical geneticists knew this was a problem, knew this was an issue, but they never worried about the clinic. And the doctors didn't understand about common variants in Africa. And so as a result, you had a bunch of misdiagnoses, patients who were diagnosed as having hypertrophic cardiomyopathy, who did not. And that's a problem because even in our top center in Boston of hypertrophic cardiomyopathy, we were misdiagnosing all these individuals retrospectively that it just had common non-pathogenic variants. They had just common variants out of Africa, and yet they were diagnosed as having hypertrophic cardiomyopathy. And what does that mean? It means you get stuck into your chest a defibrillator, and all your relatives are checked for this, quote, autosomal dominant disease. This is not fiction. This actually happened. And guess what? This is in our best hospital. I can tell you, because I've been in startups, there are many commercial labs that are still reporting out these variants as being pathogenic. So there was no talking between the stat gen community and the clinical genetic community. And at first we didn't want to publish it because we thought everybody would know this because we knew our stat gen community. But then when we looked at the clinical community, they did not know it. So you've got to make that difference. And that's why we were able to publish in the New England Journal of Medicine, we published a paper about this. But this is just the tip of the iceberg of the disparities that are happening unintentionally. So we're not trying to discriminate against African Americans. We're actually trying to help them, but we're actually hurting them unintentionally. And this was done by Raj Manray, that PhD in bioinformatics and integrative genomics. Next of the 5 lessons, the third is pitch in individual cases where standard medicine has failed. Now, these cases are interesting because when standard medicine has failed, it's a very different ethical landscape than when standard medicine works. For example, type 2 diabetes, it's not great to have type 2 diabetes, but we have medicines that sort of work. If you take care of yourself, if you do all the annoying things that we have to do every day, which none of us like to do, you could probably live a normal life other than being very busy and worried about it. But and doing anything else, like trying a new drug, like if one of us decided to try a new drug, that'd be crazy because I might poison you. Drugs are poisons. But if things are going to hell in a handbasket, if the patient's going down the drain, it's actually a very different moment and a huge opportunity. And it's where computational biologists can have a huge impact. Let me give you a story to illustrate this. A true story. It's actually the story of a son of a friend of mine. My friend is a professor of pediatrics at Harvard Medical School. He has access to all the resources at Harvard Medical School and Children's Hospital. His child, now several years ago, when this child was 3 years and 10 months old, had a bloody stool, bloody poop, and he was diagnosed with something called ulcerative colitis, inflammation of the colon. And when they did the endoscopy, which is they put up a scope up your rectum, very pleasant, and it showed that it was colitis, that was inflammation throughout the colon. But they put him on something called sulfasalazine, a very mild anti-inflammatory. The kid did great. And then when the child 10 years later goes through puberty, as often happens, and for reasons that we don't understand, interesting systems biology question, the disease flares. And the colon flares, and this child is now going to the bathroom to poop once every hour with blood. And he was a student at Brookline High School, very near the hospital, doing great. Now, if you poop every hour, not only is it embarrassing to go to school, because you have to poop every hour, but you're not sleeping at night, so you can't go to school. These kids lying in bed, and my friend saying, they try everything. They try pulse steroids, antimetabolites, first generation monoclonal antibody, fancy antibiotics that you have to get the blessing of the Pope to get, second generation monoclonal antibiotics, nothing works. So he finally comes to me and he says, Zach, I think we're going to get the colon taken out in 6 weeks because that's the only thing that works, so it's surgery. Now, when you're 13 years old, there's a number of reasons, as my late mother would say, that it's not good to have your colon taken out. A, When you go in with the surgeons, you know what you have. You don't know what you're missing when you come out. Second, that's my mother. Second is, even if you take out the colon, there's where it hooks to the rectum, there's still a little bit of tissue. It's a pouch, and that can still be inflamed and cause pain and blood, and you still will have to treat it with drugs. And third, there's the psychosocial thing. Remember how tough it was to be a teenager? Think about what it's like to be a teenager, even if you have to go around with a few weeks with a bag with poop in it, and then go through three operations to reconnect everything. It's a big deal, so you don't want to do it. So my friend comes to me and says, Zach, I saw you do that crazy presentation a few years ago that you seem to be about using drugs in silico to predict what's the right drug. Can you do this for my kid? So we have 6 weeks to go. So what crazy presentation had he seen a few years ago? So this, first of all, well, we'll get to who those pictures are in a second. This was a study done on around 50 kids presenting to a children's hospital with inflammatory bowel disease. Presenting with blood or pain, with blood and pain blood in the stool. And then they get scoped, and the way the diagnosis is made, you look under the microscope and you say, I see the inflammatory cells, does it look inflamed? And then based on that, you say, inflammatory bowel disease, which is either Crohn's disease or ulcerative colitis, or nothing. The kid's fine. And so what we did that was different, we took that tissue that he took to the microscope, and we just took the whole tissue, not single cell sequencing, the whole tissue, and doing bulk RNA expression. And this is the PCA, principal component analysis. These are the first two principal components of the PCA. And what you see, that's not too surprising, is that the kids who ended up being okay, had a pathological diagnosis of not having inflammatory bowel disease, shown in red, are over here. And the ones who had inflammatory bowel disease, shown in red and green, are over there. So what it tells you is, in this inflammatory process, no surprise, because there's inflammatory cells. I would be shocked if this is not true, that in the first two principal components, accounting for, it looks to me like 50% of the variance, you can see difference between healthy and unhealthy tissues. But we cannot separate Crohn's disease versus ulcerative colitis in the first two principal components. So that's not interesting. We never published this, although I think we're finally going to publish this. And then we didn't publish the following, which is we said, let's take in silico the studies done by one of my former interns at Children's, who's now at the Broad, Todd Golub. He created something called connectivity map, CMAP. And it's what they did is they exposed literally thousands of cell lines, the same cell line, against thousands of drugs. So it's parallel measurement of expression, the perturbation of expression across thousands of drugs in cell lines, not in our patients. So it's purely in silico. But for reasons that I'm glad to go into, is there enough similarity between the cells of the gut of this kid, or these kids, and those cell lines to say these drugs would have the following perturbation? Because what we're going to ask is, if you imagine a hyperplane through the gene expression, and I'm showing you now only the first two dimensions of that hyperplane between healthy and unhealthy, and ask yourself, what's going to take to identify a drug that pushes the transcriptome of these patients towards healthy? Again, a lot of assumptions that I'm glad to talk about later about why that's a good thing. But just like lowering glucose in diabetics helps even without understanding why that's good, shoving the transcriptome towards healthy, it's good, maybe. And we were able to show, for example, that drugs that are known to work well for inflammatory bowel disease, like azathioprine, push the healthies, the disease versus towards healthy. And we were able to show, news to me, that a drug, for example, that people use for acne, Accutane, isotretinoin, pushes the healthies towards the six. And it turns out that colitis is a known complication of this drug. And so my friend asked, could we actually do this for his kid? So within a few weeks, we got the transcriptome from the last biopsy, analyzed it, and did the analysis for him. And the reason I mentioned those guys, so the analysis was done by 3 PhDs. I wish I could say I did it, but I've gone a lot slower in coding in my older age. And one of them is, at the bottom, is one of Bonnie Berger's former students, Bonnie Berger over there from MIT, and that's him right there. Anyway, so they come to me and they say, Zach, the top ranked drug is indirubin. I said, what the heck is indirubin? And they said, I don't know, you're the doctor. And so I do what you should do, which is I Google it. And it says it's this purple thing that's part of a purple collection of metabolites called Indigo. And forgive me, for those of you who are Asian or Chinese, forgive me if I massacre the pronunciation, known as Qing Dai. And then I look into literature, and I do it knowing that you can always find a good effect of a chemical in a publication somewhere. So this totally does not impress me. But nonetheless, and in fact, this is frankly a third-rate publication. I'm sorry if you've published in this journal, but in any case, these people say that this Qing Dai, this supplement, worked for intractable ulcerative colitis. So then I call up my friend, this professor, and I say, I'm sorry, we did the analysis, and the top of this, Indigo is the top result. And instead of him saying to me, Zach, what the hell is Indigo? He said, Zach, that's really interesting. I said, why is it interesting? He says, because I had just been, he's been asking around the world about his kid's case, and there was a group in Israel, in Tel Aviv, that said, we use Indigo on all the kids in addition to the Western medicine stuff. But he had dismissed it because he's not going to give his kid a supplement. But now that I've come to him and I've told him about Indigo, he actually decides, well, maybe we should use it. I said, he says to me, where do we get Indigo? Because supplement, variable quality, depending where you buy it. I said, it's 2009, then it was 2016. Get the Israelis to FedEx it to you. And they did. And we gave it. And within 2 weeks, this kid who was pooping every hour went down to 2 to 3 bowel movements a day. Not perfect, but 2 to 3 bowel movements a day. And he's, till today, I just checked with him before the talk, kid's still fine. No surgery. And we were down to 2 weeks before surgery. The mother had given up, the father had given up, they had resigned themselves, and they tried this. And this is something that medicine could not have done. The medicine cannot do. But we have computational biologists who threw themselves into it and said, here's a case where we can make a difference. There's nothing to lose. This kid's going down the toilet, literally, if we don't do something. This is a huge opportunity, and in the Undiagnosed Disease Network, we see this a lot. All right. Become a leader. So it's not only by asking the important questions, and I know this is boring, but it's important. It's what I tell my young faculty and postdocs. Get funding and teams to answer them. I think that if you want to make a difference, it's very nice to be a methodologist and to have no one bother you while you're computing. And I love being in the zone when I'm programming, but I never get it anymore. Too bad. If you want to be impactful in medicine, you're going to have to allow yourself to get outside that wonderful zone of being a methodologist and get funding and get a team. And let me give you an example of this. This was a study that was done, published in 2009, which means the study was actually done in 2007 by one of my postdocs, Ben Rice, who MIT undergrad, course 6, went on to get a now I forgot the name of it, fellowship that allowed him to get a PhD in computer science in Cambridge, working on mutual information in music, then joined me for a postdoc, applied to medical school, got into Harvard Medical School, delayed it 1 year, 2 years, 3 years, and never went to medical school. And he took all of the emergency room data from Massachusetts, the entire state of Massachusetts. And for each patient, clumped their discharge diagnoses into these categories that you see here. And the center of that circle is when the person first shows up, As you get nearer the present, you approach the circumference. And I want you just to think, what's the diagnosis of this patient? And by the way, don't feel bad if you get it wrong, as I'll tell you the diagnosis shortly, but I've asked this question over 300 medical audiences, and this they never get right. So actually, the diagnosis of this patient is domestic abuse. And Ben Rice did this analysis, and using a simple naive Bayes predictor, was able to predict domestic abuse on average 2 years before the healthcare system was aware of it. This is in a state of Massachusetts where you're obligated to ask every patient, do you feel safe at home? But of course, and we do ask that question, but we just say, do you feel safe at home, and move on. In a 30-minute visit, you don't have a chance to really say, but are you really safe at home? But this has good enough specificity, a good enough PPV, so you could actually spend some time and get some good results. So this guy who really started working with the data, who understood the data, his name is Ben Rice. He worked with mutual information on music. He worked with me on mutual information on gene expression. You see here impossible to see, he's with Michelle Obama at the White House, working on because he was getting an award for a Facebook app to reduce obesity. And this guy has 3 R01s from the NIH. And he's working on suicide prevention, suicide detection, working with medical teams. This is a computational biologist, MIT grad, PhD in computer science from Cambridge, but he really got both the funding and the team, so he now can answer lots of questions, and I've just shown a fraction of his publications. So I'm now just going to close by saying, this is an advertisement, really, for a project that we're doing. It gives it a chance for you to get involved. We're in a very fortunate era where we have a new set of cancer therapeutics, several new sets of cancer therapeutics. But guess what? Ultimately, most of them don't work. They work for a few more years, and then they don't work. But there are individuals who are statistical outliers, even in trials that failed. In trials that failed, there are individuals who are 4 standard deviations outliers in terms of their outcome. And there's incredibly important biology there. Just like in the HIV patients who were HIV positive but never developed AIDS. And we found out the receptors that actually caused that HIV positivity to not turn into disease. We're seeing the same thing in the undiagnosed disease network, finding these rare patients who are not diagnosed. We're discovering new biology. Similarly, in this patients, we are very hopeful. And so what we're doing is we're measuring everything on them. And these are very rare patients. And we're measuring everything from the microbiome, germline DNA, somatic transcriptome, T-cell receptor profiling, and all the clinical data. And because we understand how the world works, we got an interview with National Public Radio, and we have hundreds of patients who have signed up for a very rare, by definition, rare phenomenon. And now we have pharma working with us, giving us their rare patients, even in trials that failed, who have actually responded well. And so my closing thoughts with you is time is a wasting, diseases to be cured. And for those of you who are interested in postdocs or other types of collaborations, please join us. And with that, thank you very much. So I see that I really, I lied. I thought I was going to give you 10 minutes. I only have 2 minutes. Do I have 2 minutes? 5 minutes. Questions, comments? Go ahead. And challenges, insults?
So I just have a quick question. So in this, nice talk, first of all.
Thank you.
In the example when you are selecting, finding drugs.
Yes.
The example.
Yes.
How do you weigh the risk of being wrong? What if you were wrong?
Yeah.
I mean, how do you?
So that's exactly what my grad students and postdocs were saying. They were very worried about this. I started by being less worried because I know in medicine how bad we are and how we actually prescribe drugs, and you'd be shocked and frightened how little data there is for many of the drugs we give you. But the way I reassured them is saying, we're going to rank through the drugs. There were some high-ranked drugs that we did not include. For example, they had chemotherapeutic drugs which were going to be invisible. So even though this kid's going down was going down the toilet. We were not going to give him drugs that are going to make him even sicker. So we rank everything by toxicity. There are also, in the CMAP databases, things that have never been tested clinically. So we also said we're only going to take FDA-approved drugs so that we know the toxicity profile, or supplements that no one cares about. So that's the short answer. And in this patient,